Okay. Hope you all enjoyed lunch. It is a pleasure to have Dr. Gwyneth Card here for an invited talk. This is a session with 2 invited talks, so enjoy. Thank you.
All right, thanks, Andrew. Thanks very much for the invitation to get to share my work with all of you today. So as you all know, to survive in a varied and ever-changing environment, an animal's nervous system has to be able to direct behavior flexibly. And as I think we've heard a lot throughout this meeting, of course, some of that flexibility is learned. But what I'm going to talk to you about today is how actually a startling amount of flexibility can be found in even innate behaviors, such that similar cues can drive an animal to enact different behaviors depending on context. And so the question my lab really grapples with is, how is it that from their wide repertoire of motor acts, an animal's able to elect to do the right thing at the right time? And in particular, we're interested in understanding how visual information is organized by the brain and then converted into behavioral responses. And we use as our model system the fruit fly Drosophila melanogaster for many reasons. You won't be surprised to hear one of them is that it has a relatively compact nervous system, so we think that we can answer our questions mechanistically at the level of identified neurons and circuits. Of course, there's also a wide range of genetic tools available in Drosophila for manipulation of the nervous system. But I think a really underappreciated reason to study the fly is that flies actually have a wide range of visual behaviors. Vision is a dominant sense for the fly. So here's just a small selection of things that flies do that involve vision. Their courtship ritual relies on vision. Let's see if I can lay that for you. Of course, the males will also fight. They use vision to navigate their landing, taking off and evading predators, crossing a gap. And then this over here is a video some of you may have seen before over here on the left. This is actually flies in a version of the Morse water maze task. They're on a heated floor, and they're actually using visual cues in their surround to locate a cool spot, which they learn over successive trials. So vision's also important, of course, for flexible learning in the fly as well. And so the approach we take to understand how these sort of visual cues are converted into action really focuses on 2 important, what I would call network bottlenecks in the flow of information. So information comes in through the fly's compound eye in the form of light, but then it has to be converted through successive stages of sensory processing in the central brain, being passed down the neck into the fly's version of the spinal cord, it's called the ventral nerve cord, where that information needs to be relayed, of course, to motor neurons and eventually to muscles in order to move. And throughout this flow, there's 2 places where the network contracts numerically, such that we think that we can pretty easily get a view across the network. And those 2 places are, first of all, where the information comes out of the optic lobe and into the central brain. There's only 100 cell types that carry this information, and in particular, a group of 20 what we call vision projection neuron cell types that are of interest that I will talk to you more about in a second. And the second bottleneck is literally the bottle through the neck of the fly going down the descending pathways. There's only 300 to 500 neuron pairs that carry all the information, the processed information from the brain of the fly to its nerve cord. And so we can really pose our question now sort of neuroanatomically, and we're interested in how these 20 cell types kind of in the upper left there, which are these visual projection neurons coming out of the optic lobe, how information is encoded across these. So what, in other words, is sort of the basis set of features, as it were, represented by this population. And then how is that information converted into the descending population? What is the representation there relative to actions of the fly? And of course, what is the organizational logic between these 2 bottlenecks? So what are the circuit motifs or way that this information gets transformed? And as I mentioned, in Drosophila, we're fortunate enough to have genetic tools, and I just wanted to give you a peek at, in particular, these 2 populations, the visual projection neurons and the descending neurons. We have nearly complete sets of genetic tools that target individual cell types. So I'm showing you at the bottom of the slide here just a few examples of fly driver lines, their expression patterns, and how localized they are to just individual cell types of either projection neurons or descending cells, in this case over here. Okay, so having this sort of framework that we're interested in, what behavior should we actually study? And we take what I would call a neuroethological approach. That is, we think if we're going into the center of the brain, the middle of the nervous system, we're going to get the most interpretability if we're grounded in studying a behavior that the brain actually evolved to do naturally. And so we've started in one paradigm with kind of this type of behavior, and that is how it is that flies evade predators. This is clearly something that's important to the fitness of the fly, and the video you're seeing here is one that we collected in collaboration with one of our colleagues, Anthony Leonardo, of damselflies collected from the Janelia pond as they do what's called gleaning or attacking standing Drosophila here on the side. So it turns out that when you look at the kinematics of these predator attacks, these damselflies are approaching the flies more or less with a constant velocity, and therefore they can be approximated by what we call looming stimuli. So a looming stimulus is just merely the 2D transformation of the sort of projection of the 3D damselfly attack. And so if we look at the size that the damselfly subtends on the fly's retina over time, that increasing expansion of that projection can be parameterized by simply sort of the radius of the subtended angle divided by the velocity of the damselfly's approach. And the reason that this parameterization is useful is because a large damselfly attacking quickly would basically create the same profile in the fly's eye as a slow damselfly, or a small damselfly approaching slowly. They would have the same L over V ratio. And so I mention this because throughout the talk, you will see L over V on the x-axis, and you can think of it as a proxy for speed. So for a given size of damselfly, since the velocity is on the bottom of the ratio, lower L over V ratios indicate faster attacks. Okay, so great. Now we know how we can approximate the natural attack of a predator, what kind of stimulus to use. Well, how are we then going to measure flies' reactions? Well, as I mentioned, we have this idea that the fly's response is going to be quite variable. And so to be prepared to quantify that, we thought we needed to be able to measure hundreds, even thousands of these responses. And so we've set up an automated behavior system we call the fly Pez, because it dispenses flies a little bit like those old candy dispensers, Pez candy dispensers. And the guts of it look like this. So there's just a normal fly vial that you put in the bottom of it. This is the same vial where flies are reared and housed. And it fits into the bottom of this funnel. Flies climb up this green funnel, and they go into a channel that's only one fly wide. So now they're single file. Now in this channel, we have a little laser and a photodetector so that we can see when the shadow of the fly passes a certain height, at which point we close this gate ever so softly, just in case we get a fly, we just merely pin it, we do not squish it. And the flies then, one at a time, emerge on this small platform, which is surrounded by water so that they don't run away. They stay on the platform. And what we have around the platform then is a spherical screen. So that now you're seeing the outside of the spherical screen. This contraption that dispenses the flies is in the middle. And onto this spherical screen, we show our looming stimuli. And the screen is such that we can actually project stimuli anywhere in the fly's visual field that we want. And what you're seeing here on the right is 2 videos of the fly, a side view and a bottom view, and we've collected these videos at 6,000 frames per second so that we can really follow all the different kinematics of the fly movement. So using this device, we're able to capture about 1 fly response per minute, and so we have 4 of these devices. Our best day, I think our record is we captured 2,600 fly responses to looming stimuli. So we can get a lot of data. And what do we see? Well, as I've been alluding to, in fact, a fly response to a predator attacking is not a sort of knee-jerk reflex where they do one single thing, but there are a variety of behavioral options from which they select. So for example, a fly might simply freeze and not move. It might turn away from this direction of the stimulus. It might sort of back up and pop up its wings in preparation to take off, or it might actually take off the ground, leaving the area. And it's not just that a fly selects any one of these particular behaviors, but in fact, the fly can compose these into a sequence. So here's a sequence in which the fly on the platform was grooming. You can see its hind legs grooming there. And then it's going to put those down in a quick freeze. It's then going to move its middle legs forward in a very directed postural adjustment. After that, it will raise its wings up and finally push off the ground with its legs while depressing its wings and taking off. And so I just wanted to kind of highlight for you that these kinds of responses to predator attacks, to looming stimuli, are actually quite conserved across species. So I think you're probably familiar in the rodent world with the work of Marcus Meister and others, and you'll see a looming stimulus at the top of the screen, a little bit of movement from this mouse right at the beginning, and then as the looming stimulus starts, it freezes. So the movie is still playing, but the mouse is absolutely frozen, just as a fly might do. And here is a really great example of even a human responding to a looming stimulus. This is, of course, from that great source, YouTube, if I can get it to play. And so what you'll see potentially here, okay, what you'll see here is a person, of course, walking down the street. You're going to see him do a postural adjustment, just like the fly did, and then push off the ground as a looming stimulus comes at him. Now, I would argue that this is probably the kind of experiment it's difficult to get approval for to do in the lab, hence we use the fly. So, just to show to you that, in fact, these sequences that the fly composes, again, are not sort of fixed action patterns. If we just look at 3 of these, so the sort of movement of its legs to adjust its posture, the elevation of its wings, and its jumping off the ground, also I think there's turning away, what you can see is that over different speeds of looming from slow to fast, the likelihood that we see the fly display any one of those particular behavioral modules varies, and they vary independently. Right? So these really look like sort of independent choices that the animal is making in terms of how to respond. And so in the sort of first part of the talk, what I'm going to try to describe for you today is our pursuit of the neural mechanisms by which a fly is making one of these particular choices, and that involves the interplay of the raising of the wings and the jump, the kind of end takeoff part of the behavior. And this project comes from initial observation we made from our behavioral data, that if you look at the time, the duration of the motor program of the takeoff, starting from when the fly starts to raise its wings through when its tarsi leave the ground, that you see a bimodal distribution. That is, in some cases, the fly does a short takeoff, and in some cases, it does a longer takeoff. And let me just emphasize, because many people are often used to seeing sort of reaction times or latencies. This is not a latency. This is the duration of the motor program, how long it actually takes the fly to execute that takeoff. And so if I now show you a sort of typical short and a typical long takeoff sequence, I hope that you can see right away that basically what's different between these two, which are synchronized to the point that the fly leaves the ground, is whether or not the fly takes the time to elevate its wings prior to takeoff. And in fact, we found that on average, the fly that does not elevate its wings has an 8-millisecond shorter motor program than the fly that does. Okay, and then before I go on to dig into the neural mechanisms, let me try to just convince you that this 8-millisecond choice is not irrelevant to the fly. So this is a plot of, on the x-axis, that timing. So on the right side, these are these are the long takeoffs, and these are the short takeoffs on the left side. And then on the y-axis, you're seeing a metric of steadiness, which is basically just the inverse of the amount of angular velocity the fly has once it's in the air. So a steadiness of 1 means it's no turning at all. And what you can see is that if the fly does the long takeoff, it's much steadier in the air than if it does the short takeoff. And I think this is probably best understood if I just show you a video in which I cut the wings off the fly, so it's sort of forced to do a short takeoff. And this is what we see. So without wings, the fly is incredibly unsteady in the air. And you can imagine that this might have a variety of costs to the animal. Not only does it have to spend energy potentially recovering from that instability, but it might lose its heading direction where it wanted to go. So given a choice, you might imagine a fly would prefer to be steady if it could. However, of course, if you can do the short takeoff, you probably have the opportunity to get away from your predator more often, and that is what this data on the right show. So in the case of slow looming attacks, you can see that there's no difference in survival rate between the long and the short takeoffs, and here we merely define survival rate based on whether or not the fly has left the platform when our looming stimulus would have reached a size of 180 degrees, which is sort of the theoretical time of contact. However, for faster looming stimuli, you can see that if you're enacting that long takeoff, you really do not have a good chance of surviving the predator, whether you have a significantly better chance if you do the short takeoff. So this choice, long or short, to put your wings up or not, is really a matter of trading off control for survival to the fly. And in fact, that is what the fly does. So if we look across speeds of loom, we can see that for slower looming stimuli, slower approaches, fewer flies are electing to use that short mode. And as the looming speed increases, more and more flies decide to use that short mode to take off, as you might expect from this kind of trade-off. Okay, so that sort of frames the first question that I want to dig into today, which is what is going on in the fly's brain when it's making this choice, and what are the mechanisms by which this choice is affected? So we benefited from many decades of work on a particular pathway in the fly when we started this, and that is a particular pathway called the giant fiber pathway. So like the famous squid giant axon or the fish Schmaudner cell, flies have a pair of giant axon descending neurons which, when activated, and in fact, when they fire a single spike, will enact a takeoff. And so this is a picture of that in red, that giant fiber descending neuron. And what we're showing here in the nerve cord of the animal is that the giant fiber is directly both chemically and electrically connected to motor neurons that extend the legs. So it's a pretty hardwired pathway on that end. But nobody had understood whether or not this particular pathway is actually being used when flies are using visual information to evade a predator. And so this is slightly older work from our lab, so I'll just summarize it in two slides. But what we did was we took the genetic tools that we and others had developed at Janelia, and here's a particular fly line that targets just those two giant fibers. We used it to silence the giant fibers and then examine how that affected not just the overall takeoff rate of the fly, but whether the fly is using the short or the long takeoff mode. So if we silence the giant fibers, what we found is that, in fact, flies still do take off. They can escape a predator just fine, but that short mode drops out. And if we now use our genetic tools with optogenetics to activate the neurons and record what the fly does, we see that, in fact, activating the giant fiber specifically produces the short mode. And so from this kind of data, this behavioral data plus electrophysiology, we put together a sort of model of how we think this giant fiber descending pathway is acting in relation to other descending neurons, which must also be able to coordinate a takeoff, since if we silence the giant fiber, the flies can still escape, to produce the behavior we see. And this is what our model looked like. So we presume that there's this giant fiber that drives just the sort of short mode takeoff. There are other descending neurons that can coordinate the longer mode takeoff, raise the wings, push off the ground. That's what we see when we silence the giant fiber. And from our behavioral data, which I'll sort of show here, we found that if you look at the timing of when the fly takes off, in either the case where we've silenced the giant fiber, so we know we're looking at the long mode, or just the short mode takeoff, so if we know where the signature of the giant fiber, that in fact, the giant fiber-mediated short mode takeoffs have a higher threshold to activation. When the stimulus is about 40 degrees on the fly's retina, that's when they enact the short mode takeoff. And when they're about 30 degrees, that's when they enact the other one. So that's sort of what's cartooned here. The other descending pathways have a lower threshold. The giant fiber has a higher threshold. And then here's how we sort of postulated that it might work. So if you have a slow looming stimulus, then you cross this lower threshold for the other descending pathways first. That starts in motion this longer mode takeoff, and you finish, in fact, the longer mode takeoff based on the motor delays we estimate from our data before you ever even cross the giant fiber threshold from the sensory point of view. Conversely, if you have a rapidly expanding stimulus, then sure, you pass the lower threshold first for the other descending pathways, but before the motor delay can elapse in order to enact the pathway, or the action is driven by those pathways, you cross the higher giant fiber threshold and you get your short mode takeoff. And of course, there's cases in the middle where you would start raising your wings with these other descending pathways, but the giant fiber would interrupt and complete the pattern. And so based on this model, we were able to sort of simulate this bimodal distribution that we initially observed in the data, and also this biased choice. So from our model, the faster the stimulus went, the more flies chose to use the short pathway. Okay, well, this is great and satisfying in a certain way, understanding the possible role of the giant fiber and the descending control, but it really sort of kicked the can down the road because, of course, it really begs the question, if what's important to making this choice for the animal, long mode or short mode, is the timing of the giant fiber activity, and it's, again, just a single spike, the giant fiber spike, relative to other descending pathways, what is it that's determining when the giant fiber spikes? And so to answer this part of the question, we were very fortunate to be able to take advantage of a new EM data set produced by Davi Bach's lab. And so we have been using this data set to go in and trace all of the inputs to the giant fiber. What is the information that the giant fiber is integrating? And so this is just to show you a little bit of what the data looks like. So we have these different round profiles that are all neurons. And in this particular data set, it's all hand tracing. So we're very grateful to our three to four tracers who've done all this work. So now in pink, you're seeing the giant fiber neuron and two of its inputs that have been traced. And so from this, what we learned is that, in fact, there are visual projection neurons, so remember that first bottleneck I showed you, that synapse directly on the giant fiber descending neuron. In fact, there's two kinds. In this case, the yellow kind and the blue kind. These are populations of retinotopically distributed neurons, and they both seem to synapse right into the giant fiber lateral dendrite. And so one of them in particular had a very compelling morphology that I want to tell you a little bit more about. And before I do that, I have to just give you a quick intro to a little bit of the fly visual system. So the fly has a compound eye, which means that the eye is arranged in multiple different facets. And that facet or columnar arrangement is maintained through the first several synaptic layers of the fly visual system. So this here is showing you in red sort of the fly's retina with each of these little compartments. And that retinotopy, that columnar retinotopy, is maintained throughout all of these neuropils. Now, by the time you get to sort of the fourth or fifth neuropil here, the lobular plate, not only do you have this retinotopy, which in this case is turned 90 degrees, so the retinotopy goes up and down this way, but you have 4 layers. And these 4 layers are getting information from a previous layer that's motion selective. And so what that means is that in one dimension, you have retinotopic spatial information, and in this layer dimension, you have information about direction of motion for the 4 orthogonal directions. There's sort of front to back motion, back to front motion, up and down. And it's into this, this retinotopic motion direction map that one of these visual projection neurons that will then input to the giant fiber sends its dendrites. And that cell type we call LPLC2. And so here's what it looks like. So, here are its dendrites that are in the lobular plate. This is just a single one. This is a population of about 100 neurons. And like all of these other visual projection neurons that I sort of told you about, it has a particular feature that its dendrites are retinotopically arrayed sort of up and down throughout the lobular plate, but its axon terminals are bundled in a particular location in the central brain. And this has been suggestive to many people that, in fact, perhaps these neurons are sort of throwing away spatial information in order to encode some higher level feature. And in this case, as I said, the morphology was very suggestive. So here are what the 4 main dendritic branches of an LPLC2 neuron look like in the lobular plate. And remember, I told you the lobular plate has 4 layers. Those layers are each responsive to a different direction of motion. The LPLC2 has 4 primary dendrites. Each dendrite extends into one of those layers. Furthermore, if we now take the orthogonal view of the LPLC2 neuron, the dendrites form this sort of cross-like arrangement such that each dendrite extends in the layer it's in along the direction of motion that's being encoded. And so this cartoon is kind of trying to show you what that looks like. And so what does that mean? You can imagine that if there were a stimulus centered on this particular neuron's receptive field, the stimulus that this neuron should be most responsive to, just guessing from its morphology, is one that was expanding outward from the center along each of these different directions of motion. And so a very talented postdoc that has been co-advised by myself and my colleague Michael Reiser set out to test this directly. And so he devised a two-photon imaging setup where he could take advantage of these genetic tools to image from individual LPLC2 neurons while showing looming stimuli that he had particularly centered in the receptive field of whichever region of interest he was looking at with his calcium imaging. And so remember, we sort of predicted that an expansion stimulus such as this one or a looming stimulus would be the signal that would most excite these neurons. And in fact, that is what we see. But From the morphology alone, and presuming simply excitation, one would imagine that motion itself in any one of the directions, so not requiring a loom, but any particular motion direction, should also excite this neuron a little bit. And that's not what he sees. In fact, he found that the LP-LC2 neurons are able to sort of reject this extraneous noise and be what he calls ultra-selective for looming stimuli in particular. And I don't have time to go into the details of that. I'm happy to answer questions or talk about it later, but let me just suffice it to say that this selectivity comes from not just the excitation predicted from the morphology, but also inhibition from other motion-sensitive layers in the lobular plate, which are effectively canceling out the inward motion. Okay, so to return to our question of what is setting the spike timing of the giant fiber. You just saw that the giant fiber gets input from two visual projection neuron types, one of which LPLC2 is in yellow. And in fact, LPLC2 makes about 1,300 synapses, this is across its population, onto the giant fiber, whereas a second visual projection neuron type, LC4, makes even more, 2,442. And it's true that there were actually 6 other visual projection neurons that made synapses onto the giant fiber, but all told, those other ones made a sum total of 12 synapses. So we really think that from our EM data tracing, LC4 and LPLC2, these 2 visual projection neuron types, are really the primary input to the giant fiber coming directly from the optic lobe. Great. What does that mean for how the giant fiber is responding during looming stimuli? Well, again, we can take our genetic tools and we can do sort of specific lesion experiments, as it were, by using our genetic tools to silence either LPLC2 or LC4. In this case now, we're not going to use calcium imaging as we did to look at LPLC2 responses. We're actually going to use whole cell patch clamp recording. And these are our 2 intrepid electrophysiologists, Katie von Rijn and Jan Ache, who performed this work. And they're able to, in a fly that's being exposed to a visual stimulus screen and is freely able to move its legs, its wings, so we could monitor its behavior, they're able to go in and target the giant fiber for recording in flies in which either of these inputs have been silenced. So as a control, as our kind of baseline, we silence a different visual projection neuron that we know does not make synaptic connections with a giant fiber, LC11, so in black will be our sort of control. And if we go in and silence those LPLC2 ultra-selective looming detecting neurons, here's what we see. We see that, in fact, the giant fiber response to loom decreases significantly. So here on the bottom is the looming stimulus. This is the size of the disc on the fly's eye over time. As it increases, you can see in our control flies, and these are average responses of many flies, you can see that the giant fiber initially has a depolarization transient and then slowly depolarizes again as the stimulus gets bigger and bigger. However, if you silence the loom detector, then in fact, much of this response goes away. If you silence that other input, LC4, then we also get a decrease in the giant fiber response, but not as much. And we did a little bit of just addition, basically, to show that the sum of these 2 neurons, plus some inhibitory elements that I won't talk about in detail today, basically can recapitulate the control response. So in other words, our EFIS confirms what we saw in the M, that we think these really are the 2 cell types that are contributing to the giant fiber's whole looming response. And the reason that that was significant for us is because it meant that now we can go in and we can use our silence data from one of them in order to interpret what we think the other cell type is encoding for the giant fiber. Let me show you an example of what I mean. So here is a case where we have silenced the ultra-selective looming detector. We use our genetic tools and we presume it's no longer in our circuit. And now, if we look at the giant fiber's response, which is this blue line, we see that in fact it very directly tracks in pink, which is simply the angular velocity or the edge velocity of the stimulus. And so we interpret this to be the contribution that the other cell type, LC4, is making to the giant fiber. And so we can quantify that particular observation a little bit more closely. If we now take our recordings from many different speeds of loom and overlay them, what we see is that the giant fiber response doesn't matter on whether the loom was fast or slow. It tracks the instantaneous edge velocity of that looming stimulus pretty directly. So this is on a log-log plot, so this is actually a linear fit. So we can do the converse thing. We can now silence LC4 to see what we think the LC2 contribution to the giant fiber is. And sort of long story short, what we found there is that, again, over multiple speeds of loom, it doesn't matter what the kinematics of the particular looming stimulus were, the instantaneous size of the disc on the fly's eye is what best matched or predicted the giant fiber's response. So we really think that these are two separate looming channels. One, the ultraselective looming detector, LPLC2, that's encoding the instantaneous size of the disc or the predator on the fly's eye, and the other one that's sensitive to its velocity. And this is interesting because there's a long history of people trying to understand how looming is being encoded in the brain. And these are two parameters that have come up often. And in fact, they're often called rho for the velocity and eta for the size. And so let me just really quickly show you across species that these observations have been made in pigeon, cat, mouse, locust, crab, fly, whether it's rho neurons encoding angular velocity or eta neurons encoding this size, and especially with that particular shape that has a peak before the end of the loom encoding a size threshold. These seem to be somewhat ubiquitous parameters. And so what these plots here are showing you, the sort of lines here, is they're showing you that one way to determine if or one way that people have looked at to see if a neuron is encoding rho or eta is by the timing of the response, of either the peak firing response or in our case, the peak depolarization of the neuron. And you can see that in the pink, you're seeing the eta lines, they tend to be a steeper relationship over the speed of the loom, whereas the rho neurons, because they're simply encoding velocity, and you can imagine as a looming stimulus increases, velocity is increasing till the end of the stimulus, they tend to be flatter. And so I point this out to you because we did this same analysis with our data. So if you imagine on this particular plot, if the giant fiber were acting as a pure eta element, just encoding a size threshold, then if we looked at the time of its peak response by loom velocity, this is sort of the line we think the data would fall along. If it were acting as a pure rho element, the data would fall along the blue line. And as you might guess from what I just told you, that we think the giant fiber sums these two, the actual giant fiber response lies somewhere in between. And of course, once more, we can check this with our genetic tools. So if we look at our LPLC2 silence data, so we take out eta and we only leave rho, then in fact, our responses lie exactly along the sort of perfect rho prediction. And conversely, if we now knock out the LC4, so we're only left with our sort of size encoder, we get the opposite response. So we really think now that we can sort of predict the giant fiber response to a loom simply based on these two optical variables, these conserved optical variables, rho and eta, again, along with just a couple small inhibitory elements that I'm not talking to you about today. And so let me just show you a quick cartoon to kind of maybe summarize for you how we think this works. So in the case of a slow predator attack, the computer will freeze. Ah, there we go. Okay. In the case of a slow predator attack, then we only really activate this size detector. It's not going fast enough to activate our velocity detector. So you get a subthreshold response in the giant fiber, and then remember I told you there were other descending pathways also receiving this visual looming information. They take over, they enact the long mode response. However, in the case of a fast predator attack, now you get both the size and the velocity input to the giant fiber enacted somewhat simultaneously. You get a single spike in the giant fiber. It beats the other information coming down the parallel pathways, and you get a slow mode response. And so, in fact, we see that this is what we think is underlying the fly's ability to choose between the slow mode or the long mode, the short mode or the fast mode, because if we take our LC4 silenced flies, so all we have left is the size detector, then the flies can still respond to looming stimuli, but they no longer show this bias in the behavior to increase the number of short takeoffs they would produce in response to faster stimuli. So they kind of lose the ability to adapt to the speed of the predator attack. Okay, and so I just wanted to, at this point, kind of highlight for you that I think this is a nice sort of worked example, because between the story I just told you and work of our colleagues who have been looking at the upstream computations happening in the visual system, this is now a case in which we can connect photoreceptor to muscle. So what you're seeing here are just from the EM volume, the data we had that went through that volume. So here we know which neurons in the medulla that are responding to sort of very local light and dark changes connect to these red neurons that are actually the motion direction encoders, which then feed into the LPLC2 neurons, which, as I told you, are the loom detectors, and finally into the giant fiber descending neurons, which, as I told you, go down directly to the motor neurons of the fly. So we now have that whole chain. But lest we feel too self-satisfied, let me show you what we also learned from the EM data, which is that in addition to these two cell types, LC4 and LPLC2, which input to the giant fiber and are the only direct visual inputs, there are actually 550 other neurons that directly synapse onto the giant fiber. So as much as we think we may have understood something about how looming is being computed and transformed in this pathway, there's clearly many other elements that the giant fiber is integrating in order to, as it were, make this decision about when to fire. And I think this highlights how even these, what might appear to be rather reflexive and hardwired pathways are going to be under broader control from other sort of more central elements in the nervous system. And let me just sort of finish up with a second story that kind of highlights this for you. So this really opened our eyes to this idea that there might be some kind of contextual modulation in these circumstances. And so the question we then posed was, okay, well, when else would flies be exposed to looming stimuli? I sort of gave you the very strong case of a predator attacking, but of course, beyond the fly seeing looming stimuli when a predator attacks, a fly might also see expansion on its retina when it approaches an object. Often this would be a desirable object that it, for instance, wants to land on, as in the case of the fly sticking out all 6 of its legs to land on its post. So the second question we pose then is, how is it that a fly, given potentially the same looming information, chooses whether to take off or to land? And just to sort of show to you that, in fact, literally the same expansion cues on the fly's retina can cause either of these, let me show you just a little behavioral experiment we did. So here's a fly now pin tethered in an arena. It has a visual screen around it, and you're going to see a video, a bottom view and a side view. So that was the first looming stimulus you saw. It caused the fly to take off. Now, the exact same looming stimulus is going to be shown to this exact same fly. There's been no cut in this video. And what you'll see is you'll see all 6 legs stick out, just like that. So the same fly, within the matter of a few seconds, decided to either take off or land in response to literally the same thing being shown on the screen. And of course, as you probably gathered from that video, the main thing that was different was sort of the behavioral context that it found itself in. Was it standing or flying? That's what influenced the choice. And so in order to try to understand what might be modulating these pathways, we wanted to get a handle on what descending pathways might be coordinating the landing response. I've already shown you that what we know about the takeoff, but we didn't know anything about any neural substrate involved in the landing behavior. And so fortunately, we were able to screen our collection of descending neurons. We did an activation screen where we simply tethered the flies, expressed redshifted channel rhodopsin in the descending neurons, and then saw what happened. And we found 2 descending neurons that when we activated them, caused the fly to do what is in fact its landing response, which is sticking out all 6 legs simultaneously, as it would if it were going to land on a substrate. And so we then did the sort of converse silencing experiment. We took those 2 lines, we silenced them, we showed the fly a looming visual stimulus. It's kind of denoted here. And so here are the controls showing you the probability that the fly exhibits that 6-leg extension in response to a looming stimulus. It's pretty high. If we silence something early in the visual system, L1, L2, then in fact we can knock out that behavior. And then we can knock the behavior down a lot if we silence these neurons, and even more so in a sort of unilateral expanding stimulus of a bar moving. So we think that we've found the descending pathways that are involved in this landing behavior. Great. What does this mean for the locomotor modulation of this decision? Well, we can then go in, again, with our electrophysiology tools. We can record from these neurons, and here's what we see. So in this situation, because the fly is in our recording setup, we're going to show a bar instead of a looming stimulus that has a similar kind of motion across half of the eye that a looming stimulus was. And you can see in both of these neurons that we think are involved in landing, we see strong responses, strong spiking responses to this sort of expansion motion when the fly is flying. If we do exactly the same experiment in a sort of quiescent fly or a non-flying fly, then in fact, what we see is that these visual responses are severely attenuated. In the case of the first one, DNP10, in fact, we don't see any spikes whatsoever. We only see subthreshold responses left. So there's some kind of gating that's integrating locomotor information about the fly's current behavioral state with this looming or expansion information in order to produce the response. And this gating holds across different speeds of bars. Okay, so then in just these last couple slides, let me show you how we think that this gating works. In the case of one of the neurons, DNP7, we can show that if we add the flight neuromodulator octopamine to the bath, then we can mimic the flight effect. So this looks like sort of neuromodulatory regulation of this descending neuron. This is showing you that we get many more spikes, or sort of the enhancement of the response, the visual response in this neuron. But that doesn't hold for the other neuron. The other neuron is integrating this behavioral information in a different way. So if it's not neuromodulation, the next thing we thought is perhaps it's reafference. And so Jan, the postdoc who performed these experiments, did a set of experiments where he took flies that were flying. So this is the normal response of DNP10 to visual stimuli when the fly's flying. And he just hacked off, one at a time, all of the different appendages that we thought might contain mechanoreceptors that could sense the fly's flight. The wings, the halteres, the antennae, and to see if we could see any decrement in this visual response that might go more close to what the fly's response was during no flight. But he saw no decrement. So our current hypothesis is that in fact there's some kind of efference copy of the flight motor that is being integrated directly into this descending neuron in order to effectively decouple it, as it were, from the visual stimulation. And I say decouple instead of turn it off because these neurons, which are in non-flight, not sensitive to visual information, are still acutely sensitive to mechanosensory stimulation. And so here's my last slide. Here's a little cartoon of how we think this is working. So we think that descending pathways are basically coupled or decoupled from sensory input depending on behavioral state. So you can imagine in the case of non-flight, here was sort of the first story I told you, you have these visual projection neurons which are potentially encoding different features about ethologically relevant stimuli such as a looming predator. They can be combined by these descending neurons in different ways to produce actions such as takeoff. But those aren't the same set of actions that you want available to you if you're in a different locomotor state such as flight. In that case, what you want to do is you want to use, what we've observed, a couple different parallel mechanisms, both neuromodulation and potentially efference copy, in order to switch which channels are available for you or sort of are connected to your visual stimulus in order to enact the appropriate behavior, in this case, if you're flying, landing. And so with that, let me just make sure that I say thank you to all of the people in the lab. It's a really great team of people that produce all this work, and I hope I showed you their pictures as I went along, and of course to all our Janelia colleagues who contributed to this work as well. Thank you.
If you're a student or trainee, feel free to cut the line.
Hi, very interesting talk. So I have a question about your stimulus, the looming stimulus. So in zebrafish, it has been shown that the dimming and the expanding edges component are processed by separate pathways. Did you check if that's also true for the fly?
Yeah, I think that's very similar to what we see here in the sense that the dimming is basically proportional to the size of the stimulus, right? The number of black pixels that are black. So I think that we could, we could check to see if these other parameters are directly encoded, but I think it's going to come through those same two pathways.
Hi, thanks for the great talk. Are you able to make the similar sort of bimodal distribution for like any individual fly, and how much like variability do you see for an individual?
That's a great question. We've done this most in tethered flies during the electrophysiology, and the answer is yes. An individual fly will do one or the other type, so it's not necessarily per fly.
Hi. Have you looked at whether behavioral state modifies more the upstream parts of the circuits, the visual pathways, or does it even modify the giant fiber activity itself, things like that?
Yeah, that's a great question. So both. So it's known from work by others, for example, Janelia, that there is a sort of a motor signal in some visual elements, not necessarily in LPLC2, LC4 that I showed you. We don't know that yet, but certainly in sort of parallel other projection neurons. And we also see signals of the leg motion in the giant fiber itself.
Hi. So I noticed in the DNP10 neurons during flight versus stationary, there seemed to be a really large shift in the baseline membrane potential. Is that real or just an artifact of how you plotted it? Could that just be some permissive depolarization that the Efren's copy signal is feeding into this neuron to just push it past threshold once it gets the right kind of visual stimuli?
Yeah, basically, I'm glad you observed that exactly as we say. I didn't have time to kind of dwell on that, but that's one of the reasons that we think that it's Efren's copy directly into the descending neuron as opposed to something presynaptic, because we do see that subthreshold signal go up with flight. Yeah.
Great. Let's give another round of applause to Dr. Gwyneth Card.